All right, everyone. Welcome to our final session of the Virtual Evolution Conference for 2023. We only have one session going on right now, and that's going to be this final session of the Hamilton Award finalists. As a reminder, this is part of the conference, and so therefore we do have our safety officer right here in the room. So anything that feels off to you, feel free to contact them either directly in the chat or through the conversation to everyone. There's also going to be a link that I will post in just a moment about that. And in the meantime, Rosalind, you can go ahead and share your screen and we'll get you started right at 8 o'clock. Go ahead and start.
All right, thanks so much for coming, everyone. My name is Rosalind, and I'm going to be telling you about how a hidden layer of achromatic plumage modulates the brightness and saturation of colorful feathers in tanagers and other birds. So birds are strikingly colorful animals, and the colors they produce span the rainbow. There's a corresponding diversity in the mechanisms used to produce these colors. So for example, in this redneck tanager, the black feathers are colored by melanin pigments, the red and yellow feathers are colored by carotenoid pigments, and the blue and violet colors are produced by nanostructures made of keratin and air within the feather. So in this talk, I'm going to tell you about a really surprising feature of this colorful plumage, which is that hidden underneath this layer of colorful feathers, many birds have a layer of black or white plumage that alters the brightness and saturation of the colorful layer. This work is mainly based in the tanager genus Tangara. So these are songbirds distributed throughout South America, Central America, and the Caribbean. And they're well known for their color diversity, which includes colors produced by pigments and structural colors. So if you take an individual feather from one of these colorful patches, you'll see that the feathers are really only colorful at the tips. And feathers are layered on the body so that there's a continuous colorful surface. But within a single feather, there's this really interesting gradient where there's a gray downy base and a colorful tip. And then there's this intermediate zone, which is black or white. This black or white part isn't actually visible when viewing the feathers on the body. So when I talk about the hidden part of the feather for the rest of this talk, that's what I mean. In the last few years, as I've been studying feathers, one really fascinating pattern that has started jumping out is that the color of this intermediate zone varies predictably between white and black based on the color of the feather tip. So specifically, red and yellow carotenoid feathers have this white zone, and then blue and green structurally colored feathers have this black zone. It's possible that this predictable gradient in coloration reflects some kind of developmental constraint in color production that has no functional role. But in this talk, I am hoping to convince you otherwise. So if we think about these feathers with their achromatic zones layered on the body of a bird, essentially what this gradient in color in single feathers means is that beneath the outermost very colorful layer of plumage, there's an achromatic layer made by the hidden parts of feathers. And because feathers are not opaque, it's possible that there are optical interactions between these contiguous layers of colorful and achromatic feathers. In this project, we're hypothesizing that this hidden layer of black and white plumage is under selection to alter the color properties of the feathers layered on top. Specifically, we think that white layers are changing the brightness of pigmented feathers. Pigments work by absorbing particular wavelengths of light, allowing other wavelengths to be reflected by the surrounding tissue. If you look within the pigmented part of a feather, there are often tiny air spaces that scatter light. And we know that this increases the brightness of pigmented feathers. We think the same phenomenon is happening on the macrostructural level of feathers layered on the body, so that light scattering from white layers back through the pigmented feathers increases the total brightness of red and yellow plumage. But something different is happening with structurally colored feathers. Structural colors work by selectively scattering particular wavelengths of light from nanostructures within the feather. White light scattering from below the feather would actually dilute this effect and create sort of like an unsaturated, washed out appearance. There's actually usually a small layer of melanin pigment deposited within the feather beneath the nanostructures that absorbs some of the light that might otherwise be backscattered. So again, we think these black feather layers are also a macrostructural adaptation to absorb light that would otherwise backscatter through these structurally colored feathers on top. And before I explain how I'm testing these ideas, I do want to mention that this kind of multi-layer color production is actually known from other systems. So for example, the dermal chromatophore unit in many animals contains distinct layers of pigmented and structurally colored cells. Hidden achromatic layers are also common in art. So for example, both painters and glass artists include layers of white gesso or glass underneath pigmented portions, and those amplify the colorful layers on top. So back to birds. In this study, we wanted to test whether this hidden layer of achromatic feathers modifies the appearance of visible colorful plumage, and overall understand the implications for the perception and evolution of colorful signals. So I'll walk you through the main research questions that we had and present results for each question. My first question was, is there actually a hidden layer of plumage made of white and black parts of feathers, and what are its properties? So to answer this question, I removed the colorful feather tips from a Paradise tanager specimen and directly measured the reflectance of these hidden layers. These hidden parts of feathers do actually form a solid and continuous layer of black and white, and the reflectance curves show that this hidden layer has white or black reflectance that's really comparable to white and black feathers that we might see in the outermost layer. Another interesting feature of this achromatic layer that has emerged from looking at specimens is that these color gradients actually change at the margins of different patches. So for example, if you take a black feather from the middle of this black patch, it's black all the way down to the downy base. But if you take a black feather from the edge of the patch where it's layered under a red tip, there is actually a white zone. So here's a picture of what that actually looks like on a specimen. On the left is an image from the middle of this yellow rump patch where I've moved the feathers so you can see these white layers sort of peeking out. And then on the right, you can see that at the edge of this patch, the yellow feathers switch to having black laces. And this is a similar effect from the junction of a blue and red patch. So these red feathers actually have white zones, except at the edge of the patch where they're layered under blue tips, and then they switch to having black faces. So the lability of these color gradients suggests that they're not just a developmental byproduct of color production, and that these hidden achromatic layers might have an optical function. So next we tested how the achromatic layer actually affects overall plumage color, and in particular, whether it has the effects on carotenoid brightness and structurally colored saturation that we predict. So I sampled Tangara feathers from museum specimens, and I focused on a set of 6 carotenoid patches and 6 structural patches. And then I sampled 3 feather replicates per patch for males and females. We experimentally measured the effects of background on plumage reflectance by placing pigmented and structurally colored feathers on white, black, and gray backgrounds. And then we measured changes in reflectance using a few different approaches. So first, we used a multispectral camera to capture reflectance of entire feather regions on a synthetic background. And our background was this grayscale color checker, which is a color standard that's often used in photography that has a known reflectance that's sort of in the same range of what we see in birds. So we took photos of feathers on these different backgrounds, and then we segmented out the background. So we have hue, saturation, and brightness for every pixel in this feather region. And this part was led by my collaborator, Jerome Ollie. We also used a similar approach where we put these feathers on synthetic backgrounds, but took the reflectance of individual feather branches, which are called barbs and barbules, with a technique called microspectrophotometry. And then finally, we took measurements with the achromatic layer itself as background, using this specimen that I previously talked about where we had removed the colorful feather tips. So we put feathers onto these achromatic cutouts to measure their reflectance using a hyperspectral camera. So in hyperspectral imaging, basically every pixel in the image is associated with a full reflectance spectrum. So from there, we can sort of define regions of interest and subsample pixels so that we end up with a lot of reflectance data. And this part was led by Ben Hogan, who is a postdoc in the lab. So now I'll show you evidence from a few of these approaches showing that the hidden layer really does change the appearance of visible plumage. So here's the reflectance of blue feathers on black and white backgrounds from the microspectrophotometer and hyperspectral camera. So these plots are reflectance spectra, which show the percent reflectance across a range of wavelengths. You can see that on white backgrounds, these blue feathers have much flatter spectra, which represents the backscattering of light from the background and overall decreases the saturation of the feather. These reflectance spectra are really similar between different methods, meaning that whether we measure the entire feather region or individual barbs, and whether we're doing it on the synthetic background or on a specimen, we're seeing remarkably consistent changes in color. So I'm just showing you the effects for blue here, but for red and yellow feathers, we're seeing similar results, except that brightness is changing more than saturation. And that's exactly what we predict based on what we know about the physics of how light interacts with pigments versus structural colors. So at this point, we know that the achromatic layers have substantial effects on plumage color. So this leads to a lot of potential questions about how this hidden layer might evolve in different contexts. But one I've been really interested in with tanagers is whether this layer is different between females and males. Tangara females and males look pretty similar, except that the females tend to have slightly darker plumage. So if there are sex-specific selection pressures acting on females and males, we might expect that there's sexual dimorphism in the achromatic layer. So I'll walk you through an example in Tangara parzudaceae, the flame-faced tanager. So here I'm plotting the reflectance spectra for male and female crowns. And these are measurements from specimens. So they represent color of plumage accounting for both the colorful and achromatic layers. You can see that males actually have brighter yellow patches than females. So what is the actual source of this dichromatism? And is it from the pigments in the colorful layer or the achromatic layer? So if we just look at the male feathers, you can see that male feathers tend to have this prominent white achromatic zone that I've been telling you about for the last few minutes. But if you look at the females, you can see that this white part is actually absent, and instead females have a gray or black achromatic zone. So this dimorphism is really striking and suggests that sex-specific selection pressures are acting on this part of the feather. But this becomes even more interesting if you look at dichromatism in the colorful part of the feather. So it turns out that female feathers are actually brighter yellow than males if you compare individual feathers on the same background. And that's not actually completely unexpected because female feathers have less pigment and pigments absorb light. But this means that greater brightness in male feathers on the specimen and on living birds is actually due to the effects of the achromatic layer. I'm showing you in this pattern in Tangara parzidaceae because it's one of my favorite tanagers, but we're actually seeing similar patterns across Tangara, where variation in the achromatic layer explains a lot of sexual dichromatism, especially in carotenoid patches. This suggests that achromatic layers are subject to sex-specific selection pressures for conspicuousness or crypsis. And overall, I think really highlights that this achromatic layer can be a major component of color evolution. So I just want to briefly mention a couple other aspects of the study that I don't have time to talk about. The first is that I'm using a bunch of microscopy to look at the achromatic feather features and understand how these parts of the feather might be specialized for light reflection or absorbance. And then second, we built a model of total plumage reflectance that includes multiple layers of feathers. And we're using this to simulate reflectance spectra and plotting those in avian visual spaces to explore the perceptual impacts of these achromatic layers, and especially to understand whether achromatic layers actually affect the perception of individual variation in pigments or nanostructures. So if you're curious about either one of those components, I'd love to talk further. But for now, in the last minute, I want to take a step back and think about the evolution and significance of achromatic layers across birds in general. So I've just been talking about tanagers up until now, but are these hidden achromatic layers present in other birds? So I've been surveying specimens in museum collections, and it turns out that many colorful birds do have these same hidden achromatic layers. So here's a fairy wren and a manakin, both with white layers under the carotenoid plumage. And here are a few more examples, including a cotinga with black under its structurally colored feathers. And the purple here is actually a modified carotenoid pigment paired with a bright white underlayer. So this shows that these achromatic feather layers are widespread, but they're not ubiquitous. There's really some interesting variation and exceptions, which I think is super fascinating and compelling to explore, especially in the context of trade-offs between color production and other selective pressures. So for example, these achromatic zones might really trade off with the downy part of the feather, which is very important for thermal regulation. Black and white feathers also have different thermal properties, so it's possible that looking at the achromatic hidden layer across elevational or latitudinal gradients might help explain gradients in colorful plumage. So just briefly to summarize, there's an achromatic layer of plumage hidden beneath the colorful surface that alters plumage brightness and saturation in tanagers. This achromatic layer appears to be an important component of color evolution and shows really striking dimorphism between males and females. And these achromatic layers are widespread across colorful birds, suggesting that they're consistently under selection for color production, but are also likely constrained by competing selection pressures for non-signaling functions. So overall, I think that studying this hidden layer of achromatic feathers in birds can lead to some new insights into different evolutionary phenomena like the origins of sexual dichromatism, latitudinal gradients in coloration, or the function and evolution of different kinds of colors. And then I'll just quickly thank all the people who have helped with this project, especially the museum curators and staff who have allowed me to sample feathers, the people who have taught me to use microscopes. I also want to acknowledge my funding sources, especially a grant from SSE that funded the microspectral photometry at Chapel Hill. So finally, thanks to all of you for listening, and I will happily take any questions now or later.
And we should have time for at least one question. So if someone has something to ask, go ahead and raise your hand. All right, Kelly.
So the obvious question, I think, is, are you going to survey all the birds of the world?
Yeah, down the line, I would love to. So far, it's really just been sort of showing this initial layer and then spending a lot of time in museum collections looking through But I will say, based on these sort of surveys so far, it is quite strikingly common, which means that it has also evolved multiple times because pigments and also structural colors have evolved multiple times across birds.
That's amazing. Who knew, right?
Yeah, there's lots of secrets in feathers.
Yeah, very cool.
All right, so unfortunately, we do have to move on to our next person, but as I said, we will have time for additional questions at the end. Ryan, you get to begin speaking in about 15 seconds.
Okay.
All right, go ahead.
All right, thank you all for coming out. Today I'll be telling you about the evolution of reproductive isolation, how this can interact with ecological coexistence, and what this all can tell us about the rate-limiting controls of speciation. So we'll start with the fact that's rather obvious, which is that both ecologists and evolutionary biologists are very much interested in the processes that shape biodiversity. However, these two fields tend to have very different notions of what these processes are and how they operate. From an ecological perspective, research has really focused on niche availability and how this shapes the ability for species to coexist. And this has been formalized in modern coexistence theory, which has taught us how a trade-off between niche differences, which we see on the x-axis, essentially it's differential resource usage, and fitness differences, which is competitive dominance between species, can shape coexistence outcomes. So what modern coexistence theory teaches us is whether or not two species can coexist depends on whether niche differences are sufficient to make up for a given degree of fitness differences. This focus on niche availability is very different from how evolutionary biologists think about biodiversity. And I think this quote from Joseph Felsenstein sums that up very well. He said, "There can be little question that competitive exclusion sets an upper limit on species diversity, but it is not obvious that this upper limit can be achieved. There may be additional constraints on the process of speciation, constraints set by genetics rather than ecology." And what Felsenstein showed in this paper was how recombination has a homogenizing role in the speciation process and acts to prevent species from diverging in the first place, set up in direct opposition to this idea that species richness is all about coexistence. So given these differences, so these differences are interesting because there's really obvious links conceptually between coexistence, which is essentially the processes that maintain biodiversity, and speciation, essentially the processes that generate biodiversity. However, despite these links, very little work has been done in sort of thinking about the interactions or thinking about how we can bridge these two different fields in the way that they think about these processes. And there's reason to believe that we're missing something as a result of this disconnect. So one example of this is a fairly recent study from Rybowski and Matute that showed that the rate at which reproductive isolation evolves is not predictive of a macroevolutionary net diversification rate. And this sort of goes against the idea that it's only, it could only be reproductive isolation kind of controlling speciation at these large timescales, which is really what microevolutionary speciation research has tended to focus on. So what could be going on then? Why could these two different rates be decoupled? Well, what Rybowski has argued elsewhere is that there's multiple possible controls to the speciation process. Before reproductive isolation begins to evolve, a population needs to initially start splitting in two. And after reproductive isolation has evolved, the populations need to be able to persist. So this idea of persistence relates to what's known as the ephemeral speciation model, which was put forth by Rosenblum et al. 2012. And this suggests that population persistence is the rate limiting control of speciation, essentially that speciation is often initiated, reproductive isolation occurs, but then for whatever reason, those populations fail to persist, potentially due to a failure of ecological coexistence. So past models of speciation tend to make assumptions that mean they're limited in their ability to assess the ephemeral speciation model and tease apart this idea of population persistence from an ecological perspective in its potential to control the speciation rate. And this is because they often make assumptions that limit the ability of fitness differences to evolve. So if we go back to this graphic of niche versus fitness differences that's standard in coexistence theory, if only niche differences evolve, then we're guaranteed to get coexistence. But there's no reason in theory that we couldn't also get fitness differences evolve and thus a failure of coexistence after the speciation event. So to assess this question, we're going to develop a mathematical model that really links both the genetics and the ecology of the speciation process. And the ecology is largely controlled by two parameters. The first one is the width, essentially, of resources in the environment. So as we see at the top of this graph, this will be cases where individuals with any phenotype are more likely to have access to resources. And the second parameter on the x-axis is the skew of this resource distribution. And this hasn't been considered before. But as we see on the right of the graph, the maximum possible fitness differences, the max minus the min of these gray lines, is dependent on the skew, and thus this can have implications for coexistence as well. In addition to this underlying distribution of resources, we'll assume fairly standard things from these speciation models, namely that mating occurs sortatively. So more similar phenotypes are more likely to mate, and that competition is dependent on phenotype difference. So more similar individuals compete more strongly. When we look at the outcomes of the model, then, I'm first just going to kind of explain this graphic before we dive into it. So red color will be cases where there are at least two species at equilibrium. And our definition of speciation, people can ask if they have specific questions, but it's somewhat complicated to go into. Essentially, it requires three things: that the species are sufficiently common, that they're sufficiently far apart in phenotype space that hybridization is very rare, less than 1% chance of mating upon encounter. And third, that any intermediate phenotypes between the potential species are low enough in frequency that genes cannot be exchanged through hybrids. The blue coloration then is cases where there's only a single species at equilibrium, but there's some additional information provided there, which is the number of time steps at which we had two species. So darker blue corresponds to longer time with two species throughout the course of the dynamics. And this dark blue is important because we start off all of these runs with a single species. So what that means is we started with one species. At one point, we had two species, at least two species. And then by the time we've reached equilibrium, we're back to one species. So what's going on there? Well, as we'll see, what's happening is that competitive exclusion is following from the speciation event. And we're just going to zoom in to one specific instance of this to understand what's going on. So I'm going to show an animation going through time. On the left-hand side of the animation, it'll say whether there's one species at the current time step or more than one species. The right-hand side is whether the two most common phenotypes are able to coexist ecologically. So from a purely ecological model, would we predict coexistence? And then a realized fitness surface accounting for frequency-dependent competition will be moving along the top of the graph. So what we see is initially we get the development of these two extreme phenotypes that are different species and able to coexist. But then we have a third species that rises up and it's able to exclude the other two. So what's going on here, right? We have a speciation event based on our definition of speciation. It's a completed speciation event. But then one of these species that's forming is able to competitively exclude all of the others. So it's possible in principle that competitive exclusion could follow from a speciation event. And this provides an important proof of concept that coexistence could provide a rate limiting control of speciation, and that the ephemeral speciation model could explain this disconnect between the rate that reproductive isolation evolves and that the speciation rate. What we'll see is that in the model as well, ecology serves as a really important constraint in a number of different ways. And I mentioned skew earlier. What we see is that with really high skew, there's never speciation even being initiated. And this is fairly easy to understand, right? With really high skew, there's always lower fitness for these right-hand side phenotypes. So they're never able to increase in frequency, and we never get a speciation event initiating. So this really goes to show how this distribution of resources, and namely ecology, can provide a really important constraint on the speciation process. And this really relates to this idea of coexistence theory about the importance of fitness differences, because ultimately that's why speciation isn't even starting in this example. But getting back to this idea of Felsenstein, that genetics plays an important role, what can we say about the role genetics does play in our model? So to do this, we're going to compare the full model that I was just talking about with sexual reproduction, as well as a second purely ecological model with clonal asexual reproduction. So this clonal model, this asexual model, is essentially turning off any potential role of sexual reproduction or recombination. So we're going to compare the number of species we see in these two models across different competition scenarios. So the left-hand graph, what I'll call a broad competition kernel, this is where individuals compete strongly, even with others that are quite different than them phenotypically. Whereas as we move to the right, we'll have a narrow competition kernel where individuals will only compete strongly with phenotypes that are quite similar to their own. So first, the case of broad competition, we see typically there's the same number of species with asexual and sexual reproduction. So what this tells us is that essentially, in this case, sexual reproduction or recombination is not altering the equilibrium outcome in a very dramatic way. Sometimes we get one more species with asexual reproduction. Sometimes we get one more species with sexual reproduction. This is a little surprising. Unfortunately, I don't have time to explain it right now, but people can ask. It has to do with a genetic constraint surrounding heterozygotes. But what happens as competition becomes narrower? Well, now the pattern starts to change. We start to see more and more species with asexual reproduction compared to sexual reproduction. So what this is telling us is these cases with narrow competition, we really have an important role of these genetic constraints in shaping the equilibrium outcomes. And what is that role? Well, it gets back to what Felsenstein showed us in 1981. This is the equilibrium for the asexual model. We have 6 species, and they're very tightly packed, especially these 4 species in the middle of phenotype space, but they're all able to coexist ecologically. So this can be a stable equilibrium. When we look at what goes on with sexual reproduction, these 4 phenotypes are too close together for hybridization to not break down that complex. So we get hybridization, and this has a homogenizing force here, and we lose species with sexual reproduction. So we only get 3 species instead of 6, clarifying this role that basically genetics is playing in altering the system. So zooming out, then I just want to bring you 2 take-home messages, essentially. One is that, as we saw, the outcomes of speciation are very much an interplay of both ecological and genetic constraints. And it's really important that we think about both ecology and genetics when we're viewing the speciation process, as both are important driving mechanisms. So this is really, hopefully, a call for further integration of community ecology with evolutionary genetics, and kind of a little bit of a highlight of why integrating coexistence theory with speciation research is important. The second point is that speciation does not imply stable coexistence. So in theory, there's no reason we couldn't have speciation followed by competitive exclusion. And this has potential implications for what could be the rate limiting control of the speciation process from a macroevolutionary perspective. So thank you all for your attention, and I'm happy to take questions if there's time.
And there should be time for at least one question. All right. So our question here is from Ranuk.
Hello, great talk. So if I understood correctly, you showed that if there is competitive exclusion in coexistence, it reduces speciation rates. My question is, in the speciation literature, there's like really strong evidence for mostly allopatриc speciation and some empirical evidence for sympatric speciation. So how do you account for this discrepancy, if I may say?
Yeah, this is a really good question. So it is certainly true that our model has set up essentially sympatric speciation. And this is in some sense because the question of, well, when do species that form, when are they able to coexist, kind of only makes sense in this sympatric context. But it's also true that for allopatric speciation, oftentimes, you might have a secondary contact scenario wherein, two populations diverge in allopatry, but then they return to sympatry.
Sorry, but I need to cut you off so we can get our next speaker ready to go. Again, there will be some additional time for questions at the very end. All right, Ariel, you can get started.
Okay, can everyone hear me okay? Awesome, and you can see now I present, awesome, thank you guys. Okay, hi everyone, I'm so excited to be here. Thanks to the Hamilton Award Committee for inviting me to be a part of this really wonderful symposium. My name is Ariel, and today I'm going to tell you one story from my PhD, which uses genomic and phenotypic approaches in wild baboons to gain insight into what maintains taxonomic boundaries despite hybridization. We typically think of evolutionary divergence as this beautiful metaphorical tree, ancestral species split to produce descendant branches that then evolve independently over time. However, some researchers have begun to suggest that evolutionary divergence is more like a web or a network. And that's because hybridization is now recognized as a common occurrence in many plants and animals. So this presents a really intriguing question, which is, how do species who hybridize remain distinct? So one potential answer to this puzzle is that behavioral barriers minimize interbreeding between species. So this seems to be the case in systems such as crows and heliconius butterflies. And in both these cases, individuals use visual color cues to mate with other individuals of similar ancestry. And then these cues that maintain species differences are underpinned by just a handful of loci. Now, a second potential answer to this question is that hybrids are in fact formed, but there are costs to being a hybrid. So this seems to be the case in swordtail fish and flycatchers, where hybrids have either reduced survival or reduced fertility. And again, this critical factor limiting gene flow between species is caused by just a handful of genes. So of these cases that we seem to understand quite well, it seems that the maintenance of species boundaries is often mediated by a single barrier with a relatively simple genetic basis. However, I think in many hybridizing species, barriers between gene flow are likely to be more complex. So first, we generally expect gene flow, if it's strong, to be constrained by multiple barriers. Second, even for traits we thought had a relatively simple genetic basis, new work suggests that most traits are controlled by many genes of small effect. And third, simple behavioral barriers to gene flow, such as color-based mating in crows and butterflies, are unlikely in highly social animals because genetic effects act alongside really strong sociodemographic factors. So studies of single barriers caused by a few large effect loci are probably not representative of hybridizing systems more broadly. So moving the field forward really requires expanding research to highly social species that hybridize in the wild, especially in systems where phenotypic data is as detailed as this rich new genomic data. And I think a fantastic system for this is gene flow between two species of baboon, the yellow baboon and Anubis or olive baboon. They're both highly social primates. They live in multi-male, multi-female social groups. And luckily, they form a hybrid zone in Kenya, which is located near one of the longest running field sites in the entire world, the Amboseli Baboon Research Project. So since 1971, the project has focused on a wild population of hybrid baboons in southern Kenya, which derive a majority of their ancestry from the yellow baboon, but they experience immigration and integration from the Anubis baboon. The project follows individually recognized animals on a near daily basis, which means not only do we have demographic and ecological data, but also fine-grained data on each individual's life history and behavior. Now, because researchers have been studying these hybrids for quite some time, we've learned a lot, but of course it seems the more we learn, the more questions we have, and particularly, there don't appear to be obvious strong barriers or costs to hybridization. But we have a few lines of evidence to suggest that there is something restricting gene flow. So first, as you may have noticed in the previous slide, the hybrid zone is narrow relative to the parental species ranges. Second, in work I co-led with Torres Bilgeles, we found that while many of our study animals are products of a recent wave of hybridization that began in the 1980s, all other individuals in our population descend from historic hybridization events that happened in the distant past. So this suggests that there's actually been distinct bouts of hybridization in the hybrid zone's history, suggesting that something's restricting gene flow from being continuous. And finally, we also detected genome-wide evidence for selection against Anubis introgression, similar to the patterns that have been described for the purging of archaic ancestry in modern human genomes. So all of these findings suggest that gene flow is restricted, but they don't tell us anything about the mechanisms that contribute to it. So to tackle this, we really needed to integrate both genomic and phenotypic data, which I'm going to show you today. So first, it's important to understand the process of hybridization over time and its consequences for ancestry along hybrid genomes. So over the course of generations, hybridization and recombination are going to produce individuals which have mosaic genomes. So they have chromosomes with stretches of DNA that alternate in which parental ancestry a given genomic region originates from. Now, based on our previous work, we know that all animals in our population are multi-generational hybrids like this. So we generated whole genome resequencing data for 442 of them, and then analyzed local ancestry patterns along their genomes. So that's whether a given locus was homozygous yellow, so 2 yellow alleles, homozygous Anubis, or heterozygous. So moving from the schematic to the empirical data, this is what local ancestry looks like for individuals in our population. Here I'm just showing chromosome 12 as an example. On the x-axis is the chromosome position, and then on the y-axis is individual, where each horizontal bar corresponds to a specific baboon. Stretches of the genome that are homozygous Anubis are colored green, those that are heterozygous are orange, and those that are homozygous yellow are yellow. Now we can leverage these changes in ancestry along hybrid genomes to identify specific regions responsible for restricting gene flow. So to do this, we leverage patterns of correlation in ancestry across different chromosomes. So typically, we don't expect to see the non-random inheritance of genomic regions across chromosomes because a basic principle of inheritance is that chromosomes assort independently. So when we observe this non-random inheritance of certain ancestry combinations on different chromosomes, it suggests that specific ancestry combinations are being filtered out by natural selection. Now, if these unfavorable combinations are when different ancestries are found together in the same hybrid genome, the resulting pattern suggests a mechanism involved in maintaining species boundaries. So for example, in this individual here, they have sites on chromosomes 1 and 2 that are homozygous Anubis ancestry. If this pattern is overrepresented in our population, such that we get this positive correlation in ancestry at these 2 sites, it suggests that individual D, which we rarely observe in our population, is being filtered out by natural selection because of the mismatch in ancestry or the negative correlation in ancestry that they have at these 2 sites. Now, this type of approach is not new. It's been used in a number of systems to identify regions of the genome that restrict gene flow. And typically, this pattern has been emphasized in the context of explaining intrinsic incompatibilities in hybrids. So cost to hybrids once they're formed. However, what I think is so cool about this approach is that these ancestry signals can actually be consistent with other mechanisms, including barriers to gene flow that prevent hybrids from being formed in the first place. And specifically, assortative mating by ancestry can generate this pattern. So in our case, we have lots and lots of yellow baboon matings and Anubis baboon matings, but rare yellow Anubis baboon matings. So individual D here is never even created. So today, I'll just focus on whether this mating barrier can explain genomic patterns of correlated ancestry across the baboon genome. So we calculated correlations in ancestry for almost 20 million pairs of sites genome-wide, and then identified those pairs of sites in the extreme positive tail of our distribution. So these are our pairs of sites that are most likely to have matched ancestry, and they represent candidate genetic barriers to hybridization. To then test whether assortative mating explains our candidate genetic barrier loci, we use the long-term field data to ask whether the ancestry combinations at our candidate genetic barrier loci predicted assortative mating. So specifically, we asked whether a given male and female baboon mated or not, as a function of several predictors that we already know are really important in dictating baboon mating behavior. Now, the novel piece of the puzzle that we added was including a predictor that captured the ancestry combinations at our candidate genetic barrier loci for each male and female in a potentially mating male-female pair. So we ran a model for each candidate genetic barrier locus pair, going locus pair by locus pair, and asked, after controlling for all these other predictors, whether a male and female with homozygous ancestry at both sites and which match that of their potential mate were more likely to mate than males and females with heterozygous ancestry or mismatched homozygous ancestry. And here's what we find. So each horizontal gray bar corresponds to a chromosome. The sites we tested for correlations in ancestry, but were not in the positive tail of our distribution, are shown by the dark gray vertical lines. those that were highly correlated by ancestry and were tested in all of our mating models are shown by the blue vertical lines. And the asterisks correspond to 69 sites across the genome that significantly predict assortative mating in our population. So this was really exciting to us, and we've done several analyses to poke at this result, of which I'll just share a few. So one thing we did was to construct a set of assortative mating indices. So this just measures overall how similar two potential mates are based on ancestry genome-wide, overall ancestry at the sites we tested, and ancestry at our mating-associated sites. We can then ask how models including these different indices perform relative to a null model without an index. And what we find is that models including indices based on ancestry at our mating-associated sites perform the best. They also outperform ancestry genome-wide. Now, we were also interested in how an index based on our mating-associated sites compared to indices based on several ancestry-associated morphological traits that we've identified, like pelage, color, and body shape. Now, these morphological traits might be potential phenotypic cues of ancestry that the animals are using to assortatively mate on. However, what we find is that indices based on ancestry-associated morphology do not do better than the null. So the next analysis might shed some light on if animals are not cueing in on these specific morphological traits, then what are they cueing in on? So we identified whether each mating-associated site was located in or nearby a gene. We then took this list of genes and asked two things. So first, we used the mammalian phenotype ontology database, which categorizes the effects of genes into broad phenotypic categories, like skeletal system, immune system, et cetera. We then asked whether our genes were enriched for specific phenotypic categories. Second, we asked whether our genes were expressed in specific tissues. And what we repeatedly find, although it's a weak signal and what we're trying to replicate, is that our sites are enriched for nervous system phenotypes like neuron morphology, as well as gene expression in the brain. So this suggests to us that the sites we've identified may be involved in behavioral modulation. So we've identified that assortative mating plays a role in shaping ancestry along baboon genomes, but how important is it as a barrier to gene flow? So my answer to that question is it's complex. So let me show you an example. On the x-axis is time, and on the y-axis is genome-wide Anubis ancestry. Here's Alex, a male in our population. He has more Anubis ancestry than the average at around 60%. He left the group he was born in and immigrated into one social group, and he had two mating opportunities denoted by the circles, where the females had some Anubis ancestry, but really not enough to match his own. He then joined a second group, and the number of his mating opportunities increased, but his potential to mate with a female of similar ancestry dropped even further. But then after a few years, the demography of his social group changed, and so did his potential to mate with females of similar ancestry. So as you can see, the potential to assortatively mate varies between baboon social groups and over time. And further, in these social groups, aspects like dominance rank, which can dictate a male's access to mates, can modify or overwhelm genetic ancestry effects. Now, while assortative mating may be a complex and perhaps imperfect barrier to gene flow, population genetic theory suggests that assortative mating matters, and it actually may be even more important than we initially realized. So Mural-Dahar and co. recently demonstrated this, where they found that assortative mating directly reduces gene flow by preventing the formation of hybrids, as we all know, but it also indirectly reduces it by increasing the efficiency of selection against integration because of the way it packages ancestry variants in hybrid offspring. And actually, using data from our population, they estimate that the rate of purging of Anubis ancestry over time is accelerated by up to 24% due to assortative mating. So overall, I think this work reveals a few key things. First, behavioral processes leave a detectable footprint along the genome. Not only does behavior facilitate hybridization in the first place, but it can also shape interactions between loci genome-wide that maintain species boundaries. Second, we've linked behavioral processes to these really well-studied patterns of correlated ancestry, which have typically been associated with cost to hybrids. So we think we demonstrate that multiple barriers can contribute to the same genomic patterns. And I think in general, a lot of genomic patterns are actually consistent with multiple mechanisms operating at the organismal level. So field data is really important to disentangle these key processes driving patterns along the genome. Now, on the other hand, we can use genomes which capture subtle signatures of selection that may not be obvious in natural populations. So in our case, we really had to integrate genomic and behavioral data to detect subtle mating cues that may not be obvious to human observers, like morphology. We're pretty excited about these results because they point to novel genomic regions involved in pretty complex mating behavior in the wild. And I'd just like to end on one final reflection. So evolutionary biology is so compelling and even poetic to me because it provides this powerful framework for understanding both the diversity and interrelatedness of the natural world. And I think hybridization as an evolutionary process further exemplifies this by simultaneously facilitating frequent gene exchange, but also maintaining species distinctness and diversity. And with that, I have so many people to thank, particularly the leaders of the Anti-Cell Baboon Research Project, and specifically my advisor, Jenny Tung, as well as our fantastic field team. I couldn't do any of my work without them. Thank you so much. And I don't know if I have time for questions, but thanks for listening.
Unfortunately, you don't have time for questions right now, but again,
okay.
we'll have some time at the very end. So please do hold on to your questions because we will have some time for our finalists to have a chance to answer them. All right, Isra, would you like to share your screen?
Hello, yeah, am I audible?
Right now, we just see your desktop background. We don't see your talk.
Okay, sorry.
Now we can.
Yeah, okay. Cool, thank you. Thanks everybody for being here on a Saturday evening. And before I begin, I would like to thank the conference organizers and also the Hamilton Award Committee for their interest in my research. I would also like to thank my lab mates and members of the Queller-Strassman lab and my advisors, Joan Strassman and David Queller. They are also looking for postdocs, so if you're interested, please get in touch with them. And many of us will be at the in-person conference and are happy to talk to people who are interested. With that, let us begin with something that we have been seeing a lot of in the 2 days, which is that there are costs and there are benefits to mixing different genotypes. And the evolution of sex is a paradigm example of this particular phenomenon. There are benefits of sex in that populations increase genetic variation, damaged genes can be repaired by recombination, beneficial genes can be incorporated in the genome. And this is something that agriculturists have known for centuries and have leveraged for improving crop yield and stuff like that. But there are also costs of sex. And as in genetic variation increases in a population, individuals become increasingly separated and reproductively isolated, not just due to physical locations, but also other reproductive barriers. And we have heard some really fantastic talks about hybrid incompatibility today, so I'm not going to get into the details of that. And of course, not to forget about the huge energetic costs of finding a mate and all the different rituals of sex that one has to go through. But what I'm interested in talking about today is the parallels of mixing that are found in sociality. So individuals can also interact with other individuals that are different than their own self. And this is something that has interested people in the social evolution for decades. And the prime thing that people have looked in social evolution literature is the costs of genetic diversity due to the potential of cheating, in that when there is a social group that performs cooperative behavior, there is a likelihood that there will be members that will try to exploit the cooperative resources without actually contributing towards its production or maintenance. And one of the examples that is from the social amoeba Dictyostelium discoideum, and I will get into its life cycle later on, but is that when you mix different natural genotypes, they perform really poorly in movement. And this was in a 2002 paper And at that time, the kin recognition alleles were not found in the system. So the interpretation they had was that different genotypes are fighting or competing in the social group to be in a favorable location. But people have found other the sort of genetic system about allorecognition ever since. But there are also benefits of having diversity within a social group. And a key example that comes from eusocial honeybees is that colony fitness increases as the mating frequency of the queen bee increases. Now, you may say that, Isra, that's just the benefits of sex. You just said that. And then I'll tell you about the asexual conditions of tumor microenvironments, and honestly, a lot of other stuff from Peter Nonak's group, starting from the 2007 when he's showed from viruses and other specific tumor examples where if you have in different genotypes within a tumor microenvironment, these particular tumors are better at creating more circulating tumor cells, which can then invade the bloodstream and metastasize into different locations. One key thing to note from the examples that I'm mentioning is that most of these examples are from populations that may have been founded with different genotypes. But a key interesting question is what happens when a clonal ancestor mixes with its own evolved descendants, because offspring are the most readily available members of your potential next social group. So that's the question that I'm interested in this talk in answering. And so there are two possible outcomes that can happen when ancestors mix with their evolved descendant populations: social incompatibility and social heterosis. And today I'll just mostly talk about comparing the mixed populations to the expected average phenotype of the ancestor and the evolved populations. So when mixed populations perform worse than the expected average, we would say there is social incompatibility. But when mixed populations perform better than the expected average, I'll call this social heterosis. Obviously, there are examples where the social group can perform worse than both the constituent populations, the genotypes, or the mixed population can perform better than either of the ancestor or the evolved population. And to ask this question, I have just the system, and that is the social amoeba Dictyostelium discoideum, which is a soil-dwelling protist. And what it does is it exists as single cells that feeds on bacteria and grows in the soil happily. But when they run out of food, they can aggregate together into a group, which can then move towards light and heat. So they are phototactic and thermotactic. And this motile slug can then eventually, when it finds a suitable location for development, develops into a fruiting body where 80% of the cells that were part of the aggregate become the spore, whereas 20% of the cells become this dead stalk on top of which these spores are held top. And the formation of a slug is arguably the most interesting part of the Dicty's developmental life cycle. And that's because it does cool things, but also it is a complex adaptive trait where multiple cells have to coordinate their movement, move together. So there's just a lot of things going on. And it's beneficial because it allows Dictyostelium discoideum, as in also we fondly call it dicty, so I'm just going to call it dicty and not say the whole name. But it allows dicty to move farther than individual amoebae can, and thereby when it reaches the soil surface, the spores can now be dispersed by small insects, birds, guts of herbivores, what you can name it. And here is a really cool video that I'll show you about slug migration. So if you see that slugs can merge and slugs can break. So it's really cool behavior, and it's kind of difficult to observe, but the key and the interesting part about dicty's life cycle is that because the unicellular and the multicellular stages are separate, we can manipulate a lot of things about it in the lab. For example, Dr. Tyler Larson, who's a postdoc in our lab, experimentally evolved Didyscoidium such that they were only growing as bacteria for more than 100 generations and were not allowed growing on bacteria, and they were not allowed to be social. So before they could enter the social stage, they were serially transferred. And he did this for 10 clones for in 3 replicates. So more than 30 transfers, you have 10 ancestors and 30 evolved lines. But what happened during this experiment is dicty experienced relaxed selection for social behavior, in that traits like coordinated movement towards light or recognizing your kin, it's not beneficial or necessary in the selection regime. So the adaptations that maintain such traits should be lost. And that makes this these evolved lines really suited for my question of interest, is what will happen when you have ancestors and evolved and you mix them together. How will these lines behave? So for my experiment, I set up these big non-nutrient agar plates on which I lay down spores with food bacteria, and I wrap these up and leave them on the bench for 8 days. And light can enter through a really small pinhole. And during this 8 days, slugs eat the food, they run out of food, they move towards the light that enters through this really small pinhole. And I did this for in 3 replicate plates, so 10 ancestral clones, and 30 times 3, so 90 evolved plates and 90 mixed plates, where I mixed ancestors and evolved lines for a particular thing. And after 8 days, I have fruiting bodies that developed where I loaded the samples. I call these the non-migrators, in that their distance of migration towards light is 0. And I have fruiting bodies that have migrated various distances from the starting line, and I call these migrators. And I've measured the number of these fruiting bodies. I have measured how far they travel by laying a grid on top of the images and counting this in image A. And I've also collected how many spores these non-migrators versus migrator fruiting bodies make. So first of all, as expected, the evolved lines do travel a significantly small distance as compared to the ancestral lines. But interestingly, when we mix them together, the mixed lines travel at an intermediate distance. And initially, I thought that this is probably because they make different number of migrators, because most of the evolved lines, let's say, don't migrate, and the ancestors migrate in the mixed lines, so you have different proportions. But when I compare the proportion of migrators within the 3 treatments, I see that it's not really very different and very marginally better in the evolved as compared to the ancestors. So that was confusing. But the most interesting result was that when I measured how many spores these migrating versus non-migrating fruiting bodies make, I found that at the level of the plate, if I add all spores, mixed lines actually perform much better than both ancestors or evolved lines, such that they show a phenomenon of social heterosis, and suggesting that there is benefits for mixing in these particular 10 clones that I have measured. So from my results, we do see support for social heterosis in the Discoidium. It can be beneficial for ancestors and their recent descendants to be mixing. However, it is likely dependent on environmental context and how much standing genetic variation is present in the ancestors. It's possible that there is a complementary synergy happening where evolved lines are benefiting from migrating abilities of the ancestors, and mixed lines are benefiting from the increased growth rates of the evolved lines.
We'll need to wrap things up.
Yeah, that's my last slide. And there are some future questions that I'm interested in that. How is this just a short-term rescue sort of a strategy, or how long does this stay? How does it affect the said kin recognition loci that I talked about earlier? And does this mean just a decreased cost of discrimination errors or actual benefits of mixing? So in a follow-up experiment, I have mixed all the 3 evolved lines, and I'm hoping I would get to talk about it in the in-person session of the conference, and I'm looking forward to that. With that, I will thank everybody, and we'll take questions at the end of the session.
Yes.
Thanks.
I do need to ask you to stop sharing your screen so Robert is able to share his.
Okay.
And then Robert, you should be able to unmute yourself and begin.
Yeah, that's good, everybody. All right, everybody, we all made it to the end of the conference. Thanks for listening to my talk on olfactory receptors in birds. So building on Rosalind's talk, birds are incredibly colorful. They have these elaborate morphology and displays, but there's a darker side to these colors. And that is, in ornithology, there's a prevailing thought, still exists today. Birds are mostly diurnal, they're often colorful, and therefore birds rely on vision at the expense of or instead of olfaction. And so this idea was synthesized by Alex Hill in a 1905 Nature paper, where he performed morphological and behavioral experiments on turkeys, concluding that olfaction was not relevant to bird behavior ecology. And this was bolstered by earlier experiments from the most famous biologists of all time, Charles Darwin and John James Audubon. So John James Audubon buried deer carcasses under the ground and found that vultures couldn't locate them. Turkey vultures.
Excuse me for a moment, Robert.
Yes.
You're not actually in slideshow mode, so we still see all of your slides along the left-hand side of the screen.
All right. Sorry. Okay. Slideshow mode. Okay. That doesn't work, right?
Well, you're not currently sharing anything right now, so.
That still doesn't work, right?
That does work.
That
and
works.
We're going to
Okay.
start your 15 minutes from now.
I clicked the same thing that I did before. All right. So should I start over or? All right. So I'll just start from here because I think I was okay. Thanks for telling me though. So that review paper was bolstered by experiments from John James Audubon. He buried the deer carcasses, like I said. The turkey vultures couldn't locate the carcasses. He concluded that they forage using vision, not smell. If you read Voyage of the Beagle, there's a part where Charles Darwin is in South America and there's a row of these huge Andean condor birds lined up in front of him and they're all chained. And he's got a piece of meat in his hand that's wrapped up in paper. And he's showing them this paper and they're not reacting. They're just kind of hanging out. And then he rips the paper and exposes the meat and they all start flapping around, desiring the meat. He concluded the same thing, that these birds are using vision in foraging and not smell. So this thought, this was the dogma in ornithology until the later half of the 20th century when behavioral evidence started getting published. So these are 4 birds that all use smell that we now know in their foraging. For example, the honey guide uses smell to detect wax from beehives, to locate beehives. And contrary to what Audubon said, the turkey vulture does have a good sense of smell, an excellent sense of smell that it uses to scavenge. The carcasses Audubon had were too rotten. So that's the behavioral level, but what about the molecular level? Well, odors are detected by olfactory receptors, which are 7 transmembrane G protein coupled receptors that sit in the plasma membrane. And an odor comes in this case, propionate in this example, binds to the olfactory receptor, induces a conformational change that creates a signaling cascade, which depolarizes the membrane, sends an action potential, and goes to the brain to perceive the odor as a smell. And to accommodate the wide variety of odors in nature, big, small, polar, nonpolar, aromatic ring, sulfur, whatever, olfactory receptors are the largest gene family in all of vertebrates, with over 1,000 in many species. So the elephant has around 2,000, dog with 800, human with around 400, and these are all a number of olfactory receptors that were for all these species, smell is a part of their lifestyle. But what about in birds? Is olfaction important in birds? So do birds have a large number of olfactory receptors? So to do this, we have to go into the bird genomes. What we do is we build a BLAST query file. We have a BLAST query file of known olfactory receptors, and we use that file to scan the bird genomes. We recover the hits, filter them for open reading frame and intact transmembrane domains and no overlap in length. We align them, build a phylogenetic tree, and it winds up looking like this. And it usually forms this stereotypical shape where each tip here is a single olfactory receptor. At the bottom here in green, we have, there are 3 subfamilies, and the bottom here in green, we have an alpha, the yellow is gamma, and the blue are these gamma C. And the gamma C are almost always the most numerous in birds. They have short branch lengths, which means their sequence similarity is very similar, very high. And they're only found in birds. They're not found in any other vertebrate or any other extant animal. So when we do this across the bird phylogeny, we did this for 70 birds across the bird phylogeny, found 8,880 olfactory receptors total, and repertoires from individual species ran from 7 to about 400. The largest repertoires were in the earliest diverging branches of birds. So for example, the emu here, as well as the fowl, chicken, and ducks, which had around 300 or so. And also rails. Rails have a very high number of olfactory receptors, the highest of all in the black rail there. The smallest, there was a small repertoire in the hummingbird here, and also songbirds in general have small repertoires, but the smallest of all in crows. Crows, we have 4 crow species here, were consistently the smallest olfactory receptor repertoires we found. So do birds have a large number of olfactory receptors? Yes, and it's dynamic across the bird phylogeny. But are they expressed in the olfactory system? So the genomic olfactory receptor repertoire doesn't tell us about where these olfactory receptors are functioning. So in this review paper of mammalian olfactory receptors, we can see the olfactory receptors on the side here and on the top different tissues. And you can see that olfactory receptors are expressed in all different tissues, heart, lung, liver, they're all present in all tissues. So are the bird olfactory receptors present in the relevant tissue? So to do this, we obtained olfactory epithelium as well as control pectoralis muscle from 4 different birds: chicken, zebrafinch, cowbird, and hummingbird across the bird phylogeny. And what we found was, if you look at this, this is the zebrafinch olfactory receptor repertoire phylogeny, where each tip is a single olfactory receptor. We have 2 columns here. The first column is the olfactory epithelium expression level of that olfactory receptor, and the second column is the pectoralis muscle olfactory receptor expression level. And olfactory receptors are not only expressed in the olfactory epithelium, but the entire genomic repertoire for the zebrafinch was expressed. And more are expressed in the olfactory epithelium compared to the pectoralis muscle, and they're more highly expressed in the olfactory epithelium compared to the pectoralis muscle for those that are expressed in both. When we look across the phylogeny, we see this pattern in all the species that I had samples from, where there are more olfactory receptors expressed in the olfactory epithelium compared to the pectoralis muscle, and they're more highly expressed. So yes, a substantial majority of the repertoire is present in the olfactory epithelium. The chem morphology or ecology predict the olfactory receptor counts. So to do this, we measured as a morphological measurement, we use the olfactory bulb, which is the brain structure where olfactory information is received. And this can be used as a positive control, has been shown in the literature to be a positive control of that species' reliance on olfaction. And we ran phylogenetic generalized least squares methods to control for the non-independence across the phylogeny. And when we did this with olfactory bulb size on the x-axis and olfactory receptor count on the y-axis, we see a significant positive correlation between olfactory bulb size and olfactory receptor count, where the birds with the largest olfactory bulbs, for example, emu and rail, have the highest olfactory receptor counts, and the birds with the smallest olfactory receptor counts, in fact, the smallest of all, the crows, had the smallest of all measured olfactory bulbs. So this is a significant positive correlation here. And when we look at an ecological variable, for example, nocturnality, we have 4 gains of nocturnality across the phylogeny: nightjars, black rail, barn owl, and the nocturnal parrot. We see that nocturnal birds have more olfactory receptors when compared to diurnal birds, showing that nocturnality is a predictor of olfactory receptor repertoire size. So can morphology and ecology predict the olfactory receptor counts? Yes, for olfactory bulb size and nocturnality. But our real question here is, do bird olfactory receptors respond to the odors? And to do this, we had to do the first ever functional study of bird olfactory receptors. Now, what we do here is we take an approach called a consensus olfactory receptor sequence. We take a group of olfactory receptors, in this case, all of the gamma C olfactory receptors from the chicken, and create a consensus sequence by aligning all of those gamma C chicken olfactory receptors and selecting the most common amino acid at each position. So in this example alignment, the most common amino acid at this position is alanine, so the consensus gets an alanine. Here it's cysteine, so the consensus gets a cysteine, and so on until you've built the consensus olfactory receptor. We then take that olfactory receptor, ligate it into a vector, transform that plasmid into bacteria to grow it up, and then transfect that plasmid into mammalian cell culture. The mammalian cells then express the olfactory receptor on the plasma membrane, and we can bombard those mammalian cells with many, many different kinds of odors. We then measure the olfactory receptor response by measuring the level of cyclic AMP produced downstream of the binding of the olfactory receptor. And we did this for many, many different odors against the chicken gamma C consensus sequence. We found that these particular odors called pyrazines, which smell like green peppers and are found in green peppers and several other vegetables, ginger as well, cause a response in the gamma C consensus sequence. So here on the x-axis, we have the concentration of the pyrazine starting from low to high. And on the y-axis, we have the response of the bird olfactory receptor relative to the empty vector control. And we see that as the concentration of the odor increases, the response of the bird olfactory receptor, in this case, chicken gamma C consensus, response increases as well for both of these pyrazines. But our goal here wasn't to look at consensus sequences, it was to look at endogenous actual bird olfactory receptors. So this consensus sequence was made from all of the chicken gamma C, which are all of the tips downstream of this node. This is the gamma C chicken phylogeny. And so we then took 4 more consensus sequences targeting individual clades within the gamma C and tested against pyrazines. So when we do this, same setup as before, concentration of odor on the x-axis, response relative to empty vector on the y-axis. We see that clade 2 in green has the strongest response to the pyrazines of any of the 4 clades. So that let us know we need to focus on clade 2. So we went back to clade 2 and designed primers that will amplify actual endogenous or native olfactory receptors from the chicken using chicken genomic DNA. So we amplified these olfactory receptors using primers designed from the genomic repertoire. And after cloning and amplifying and testing many of these, we found one that is within clade 2, endogenous chicken olfactory receptor, that responds to the pyrazines. And this is the first match of a bird olfactory receptor to an odor, strongly indicating that chickens are capable of detecting this green, what we perceive as like a green pepper type smell of the pyrazines. So also this chemical 2-isopropyl-3-methyl-oxypyrazine is also found in the chemical communication of a variety of different types of insects. And so we hypothesize that perhaps the chicken is using this in its foraging behavior. So do the bird olfactory receptors respond to odors? Yes, the chicken endogenous or olfactory receptor sequence responded to pyrazine. So in the future, I would like to characterize the odor responses of other endogenous gamma-C olfactory receptors, as well as characterize the olfactory receptor responses from a variety of other bird species, including that turkey vulture and the honey guide. And I would just like to acknowledge my committee and my advisors, the sequencing and the funding for this project. I could take any questions.
And we can certainly take 1 or 2 questions for Robert before we open it up to everyone. All right, so Kelly.
Wow, Robert, I grew up learning that birds were anosmic, so you just changed my worldview here.
Yeah, that's what I'm saying. This is the prevailing thought. Yeah,
That's crazy.
absolutely.
I did notice, I did notice when you were showing the transcriptome of the liver and the membrane, whatever it was, that the chicken was different.
Oh,
what was
yes.
going on?
Yes.
What's
so
going on there?
Yeah, so this is a problem with the slide a little bit. There's so many olfactory receptors, it's hard to see here. But also there was one olfactory receptor, it's a gamma down here in the chicken, that's really, really highly expressed, which just creates this larger scale. And so that I should change the figure. It unfortunately makes more things yellow.
Okay.
That
So
otherwise
it looked like the chicken had no right.
Yes, it's due to
repertoire
that.
expressed. Okay, I got it.
I should change that. Yeah, I should remove
Yeah,
that one.
you just changed the scale. Okay, cool. Thanks.
Yep.
Awesome talk.
Thanks.
All right, Daniel.
Yeah, absolutely. Awesome talk. Very, very interesting. I was actually kind of thinking about future studies. Could you essentially, if you had enough data to kind of create a library of prey to bird connections with olfaction, could you actually kind of develop maybe like a barcoding effect where you could actually look at coevolution of the animal from the bird perspective to the prey, and then maybe even take that one more step further and look at global distribution? I mean, like, what are the big like in things you would like to do with this?
Well, I haven't considered that as a big end thing. That's not to say it's not valid. I think that's a great idea and would really push the envelope of what we're learning about the evolution, like arms race type thing of these receptors. For me, one of the end goals is to see, can we use these olfactory receptors to the responses to our advantage. I'm sort of suspecting that there's certain niches, particularly the turkey vulture, that are extremely sensitive to be able to detect those odors from so far up at a height where they're soaring. And if we can determine how sensitive they are, can we develop technologies that are similarly sensitive in detecting odors? So that was my idea for the end goal. I think your goal is also really valid. But yeah.
All right, at this point, Robert, I'm going to ask you to stop sharing your screen, and we're going to open this more generally to questions for these final 5 Hamilton participants. So Ben, you can go ahead and ask your question.
Hi, my question is for Robert, actually, and was related to that slide that he just took down. I'm curious about that huge diversification in that gamma C group of olfactory receptor genes. Is that a pattern that you see, do you think, in groups other than birds, or does it indicate something special about the evolution of olfaction in birds? What's going on there?
Yeah, we think it's very special. So they're not, the whole subfamily is not present in any other organisms besides birds. It's not present in crocodiles or alligators, the closest living bird relative. We think it's something very special with how birds are detecting odors, especially because we were able to link an odor to a gamma C olfactory receptor in the talk. But one thing that I didn't mention was that if you build an olfactory receptor phylogeny of multiple species, the gamma Cs, the gammas and the alphas will group in a normal or orthologous relationship 1-to-1, but the gamma Cs group according to species. So you'll have a large clade of, say, chicken that are all together, and a large clade of zebrafinch, say, that are all together. And so these are evolving in a very strange species-specific pattern. And we think that that has something to do with how birds have evolved the ability to smell. So I definitely think there's something very special in birds that they're using with this family that allows them to have a unique sense of smell in that way.
Huh, maybe a high rate of gene duplication or something that is driving that diversification within gamma C.
Yes, we think it's either gene duplication or maybe a concerted evolution, gene conversion, something along those lines, or both even. So
that's really cool. All right, well, great talk. Thank you.
Thank you.
All right, we have a question from Sarah, so go ahead.
Hi, thanks to everyone in this session. It was really terrific. I would like to go back to Brian's talk. And I was just curious, Brian, you are modeling some very large processes together in these models that you've created. And I wondered if you have plans for or have thought about using historical speciation data or experimental methods to validate some of the results that you're seeing.
Yeah, I mean, I've certainly thought about it. It's not really my area of expertise, but the thing that I think I really would like to get out of this, and if anyone has any ideas, I'd be really happy to hear them, would be, okay, so we have this proof of concept that either coexistence or just the buildup of reproductive isolation could limit the speciation rate, and actually getting at, well, what is it, and how can we actually test what it is in using real data is extremely challenging, and to me that's kind of like the $1 million question that the model opens up. So yeah, I mean, I've thought about it. I don't have any great ideas, but I'd be really happy if anyone else did to hear about them.
All right, we've got a question from Peter.
Hi, this is for Israt. So the dictee has the solitary part of its life history, right, where the individual amoeba are floating around by themselves. Are you sure it's completely solitary? So if you, in fact, put together several lines of different clones or that were genetically different, might there be some interactions that are going on in the supposedly solitary part of their life history, and might they then have a more heterogeneous population that could form, the slugs in a different way? Thank you. Enjoyed your talk.
Thank you. Yeah, I've enjoyed many of your papers. So that's a great question. And I think there was a paper 2019 or sometime where they had shown collective feeding in dictee's individual growth stage, but they had shown it in sort of clonal populations. So in general, dictee has a lot of heterogeneity in terms of either the kind of like stochastic processes, like what kind of cell stage the cells in a slug are, or what is their feeding sort of condition. A lot of processes determine where in a slug you could be, but that's an interesting thing, that mixing them in asocial conditions. Usually what people have done with, let's say, mixing mutated, like the xenic lines that are not primarily wild clones. My research is primarily with the wild clones that people have isolated from field, and not the genetical mutations in the liquid cultures that people use for developmental studies. In those systems, they have seen either sort of a competitive exclusion where one clone that grows faster sort of replaces the other. But I haven't tried that with wild clones, but they, in a maybe stage, you do see sorting. So even if you do mix them, they sort primarily into whatever genotypes they may be, and they can come back together later or sort back again later, but the specifics are a bit blurry. So thanks, that's a great question.